introduce the microbe research conducted in Fukusaki Laboratory. Nowadays, the advancement of molecular genetic technology allows many researchers to be able to engineer various kinds of microorganisms in order to produce desired products. Listed here are several efforts that have been made to engineer host organisms for bioproduction. This includes E. coli, Saccharomyces cerevisiae, Acetobacter pasteuris, and also cyanobacteria. However, bio-based production still remains a challenge. One of the biggest problems is how to further improve bio-based production to be capable for commercial production and comparable with chemical synthesis and petroleum-derived industries. In order to improve the production, the most common approach for selection of target gene is by using rational selection, whereby the gene targets are chosen based on genetic knowledge. But this approach sometimes results in unexpected cellular behaviors. On the other hand, metabolites are the final readouts of biological system representing closest correlation with phenotypic features. Therefore, deeper understanding of the metabolic state in the microorganism provides valuable information that can be applied in numerous ways. Metabolome analysis can identify possible rate-limiting step and competing pathways that can be targeted for strain improvement. Shown here are analytical instruments used for microbe research in Fukusaki Laboratory. Please feel free to pause the video and read these individually. Now, I'll introduce some research activities in the microbe field. Previously, we had several research collaboration projects with these universities. And here are examples of some of the research topics. So from here, I will introduce examples of 3 microbe research topics. The first collaboration research is with UCLA. The objective of this research was to improve 1-butanol production using metabolomics approach. So 1-butanol is important for chemical feedstock and advanced biofuel, and 1-butanol production using microorganisms is a promising way for a future industry. Usually, for 1-butanol production, Clostridium species is used, but due to the problems here, obtaining high production efficiency in Clostridium species was difficult. So, in this project, instead of using Clostridium species, the attention was focused on E. coli and cyanobacteria because it is easier to modify the genes in these organisms. So, the table here summarizes the previous studies for these 2 microorganisms. As stated here, strain improvement for commercialization requires knowledge on metabolic strain. Specifically, for strain improvement, the first step is to identify the rate-limiting step related to 1-butanol production using metabolomics. Then, the rate-limiting step is adjusted by knocking out the specific genes. Identification of the rate-limiting step and adjusting the rate-limiting step is repeated until it finally leads to the increase of 1-butanol production. Next example microbe research is with Kyoto University. This study focuses on hypothermophilic archaeon that produces useful proteins. However, the problem with this bacterium is that although the entire gene has been sequenced, we can see that function of half of these genes is still unknown. Therefore, the purpose of this project was to understand the gene function of this microorganism. Metabolomics approach can be used to identify the metabolic change based on different carbon sources, and the metabolome change can be used to hypothesize a possible gene function. Finally, the third example research is with the University of Manchester. This study focuses on streptomyces, which can produce antibacteria as shown here. Streptomyces is used as model organism to study the genetic and biochemical basis for production of bioactive compounds. And shown here are 3 antibiotics that streptomyces can produce. And for the reasons listed here, streptomyces is considered to be suitable for antibiotic production. Firstly, There are various strains of Streptomyces, as shown here, but there are some problems with each of these strains. Therefore, the purpose of this project is to conduct higher antibiotic-producing strain for each type of antibiotics that was shown in the previous slide. Lastly, listed here are some of the recent publications from our lab. This is the end of our introduction, and thank you for listening until the end.